It's a pleasure for me to be able to speak to you on Inventors Day. I thought I would just tell you a little bit about myself and some of the things that I've done. I had the great pleasure of being at Cornell from 1966 to 1970, and then later on being on the board of trustees, and even had two of my children go to Cornell. After Cornell, I went to MIT, and when I finished, I began working in a hospital. And if we could go on to the next slide, the project I began working on was to see if we could discover molecules that could stop blood vessels from growing to tumors. And and this is just a picture of why that's important, because when blood vessels grow to the tumor, they may enable the tumor to get much, much bigger. And if you go to the upper right, actually spread through those blood vessels. That's a process called metastasis, which can kill people. When I started working with my advisor, Judah Folkman, there had been really was no way to study blood vessel growth. So we had to actually invent a way to do it. And if we could go to the next slide, what we did is there's certain places in the body that, like the eye, the rabbit eye we used, also the chick-chorial and toque membrane, that have no blood vessels or very few. And so our idea to study this was to put a tumor there, and then a tiny little particle, like a nanoparticle or microparticle, that could deliver the large molecules that we were trying to isolate. No one had ever done that before, and the scientific literature and scientists in general would say, what's on the next slide, that if you tried to put a large molecule in a tiny particle, like it was encased in that tiny particle, it just couldn't get out. It was just too big, like walking through a brick wall. That was the conventional wisdom at the time. But if we were going to solve the blood vessel problem, we had to solve this problem. So I spent 2 years working in the laboratory, experimenting with different techniques, and I found over 200 different ways to get this to not work. But eventually, on the next slide, we were able to make tiny little particles, like you see a picture of one on the left and cut in half on the right. And we showed in the journal Nature, next slide, that you could use this for the very first time to release all kinds of proteins and even nucleic acids. This was the first paper ever showing you could take tiny particles and deliver nucleic acids. It's a 1976 paper. Nonetheless, when I presented this work, again, it met with a lot of skepticism. Some of that's shown in the next slide. And people would criticize the work and say they couldn't believe it because of the large molecules couldn't get through the materials. And they also said the solvents I was using, like methylene chloride and ethanol, would destroy whatever we put in. But we kept persisting. I had done a lot of this work at Boston Children's Hospital with Judah Folkman, who was my advisor, and then I would continue it at MIT. Now, because we had, in spite of that skepticism, maybe because of it, we just kept going. My first 9 grants were rejected. I couldn't get a job in a chemical engineering department. Actually, I ended up in a nutrition department for a while. But because we had this approach of having the nanoparticles or microparticles, and we developed the bioassay, we could study for the first time whether we could stop tumors from growing. And I'm going to show you a bloody slide, but there's no other way to show it. So we published a paper in Science in 1976, next slide, and it showed for the first time that you could stop blood vessels from growing. What happens normally, and hundreds of fractions we tested on the left, and they all had these sheets of blood vessels growing over them. But one of them did not. We repeated the one on the right 20 times and noticed the giant difference in vascularization. And that would ultimately lead these techniques many years later, because of great work at Genentech and other companies, to what's on the next slide, which is a summary of all kinds of angiogenesis inhibitors, blood vessel inhibitors. Avastin, for example, is the number 2 or 3 best-selling biotech drug in history. And there are many of these drugs that are now approved. And these are really the only ways to find molecules, drugs like Eylea and Lucentis, that can prevent diseases of blindness in the back of the eye, like macular degeneration. Now, going back to the 70s, Dr. Folkman and I, we wanted to get a patent. Children's Hospital never had one before. We filed for one, and 5 years in a row, it got rejected. And everyone told me to give up. But I told you in the beginning that when we first did this, everyone said it was impossible. It could never work. And I wondered, maybe somebody wrote that down. So after 5 years of rejection, I did what's called a science citation search, looking back at our 1976 Nature paper to see who wrote anything about what we said. So I found actually some interesting things. And one of the articles I found, next slide, stated the following, describing this very young field. I'll just read some of it to you. They said the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One wouldn't expect that macromolecules, for example, proteins, could be released because of such a technique, because of their extremely small permeation rates. However, Folkman and myself have reported some surprising results that clearly demonstrate the opposite. So I showed that to the people at the hospital, and they showed it to the patent examiner, and he said, gee, he had no idea. But he said he will allow this patent if I could get written affidavits from each of these scientists on the bottom that they really wrote that. So I was a young professor. I wrote them back, and they actually said that they did write that. And on the next slide, we got this very broad patent, the first time on controlled release of macromolecules. And we used that and other things we did, next slide, to, and I got involved in starting companies and teaching companies, and it would lead to all kinds of new products. We worked with Takeda, for example, and taught them how to make these, and they came up with what's called Lupron Depot, which is a product that's still widely used today to deliver a peptide to treat advanced prostate cancer and endometriosis. I think, and many others you see up here, like Risperdal Consta, which is a way that many millions of people with schizophrenia would get treated, and Vivitrol, which is a once-a-month treatment for opioid addiction, and many, many others that you see here. And not only has it been useful in medicine, it really helped because of Yanni Zohar, one of our visiting scientists and now a professor in Baltimore. This whole area of aquaculture you see in the bottom right, because for the first time you could, control spawning and even preserve rare fish. Now, one of the other things that this would lead to many years later, if I could go to the next slide, was the idea that you could not only deliver large molecules and like proteins and peptides and protect them, but maybe you could deliver messenger RNA. So the idea, as you probably know, is DNA makes RNA makes protein. By 2010, people had 70% of the top best-selling drugs were proteins. And people had done work on RNA. There was a famous paper back in Nature in 1990 showing that you might be able to get RNA to make proteins in animals, but people would find that RNA might cause different negative effects. But there was an important paper by Carrico and Weissman, who won the Nobel Prize last year in medicine for this work, showing that if you modified the messenger RNA chemically in certain ways, those effects would go away. They actually wrote a patent, and they tried to start a company, but nobody would give them funding because they didn't think this was possible. A few years later, Derek Rossi, who was a professor at Harvard, actually began to use these techniques to create iPS cells. This is a certain stem-like type of cell. And he also was interested in starting a company. He did this with messenger RNA. And so he tried to get funding and went to some very good places like Third Rock. And again, they told him this, could never work. And then he came to see me, and I thought, well, actually, I think we can get it to work, largely because of these inventions we had made. Not only the idea shown in the Nature paper that you could deliver large molecules, but we had actually taken it further. We'd come up with ways to coat it with with polyethylene glycol, a paper we wrote in Science with Roxandra Graf. We'd come up with ways to create really what are called ionizable parts of lipids or polymers. Actually, this work was done by Dan Pack and who's now a professor in Kentucky, and actually David Putnam, who's a professor at Cornell in the year 2000 and again in 2001. And so, but then to even take these ionizable materials, you want to have a lot of them. And Dan Anderson, one of our postdocs, now professor at MIT, and David Lynn came up with ways to make libraries. And then we even came up with ways that Rohit Karnik in our lab, now professor at MIT, did by using microfluidic procedures to make these. And I thought that was scalable. So my feeling was that we could certainly make messenger RNA therapeutics by putting them in the right kind of nanoparticles. So with Derek, and I talked to Nubar Fayan, who's a MIT graduate and started Flagship Ventures, about starting a company, and we started this company, Moderna. Ken Chen was also involved, who was a cardiologist, and that was really the idea, that you could have messenger RNA therapeutics. The next slide just shows that you can actually treat diseases you could never treat before, like use our mRNA to make new intracellular or membrane-bound proteins. And the next slide really shows something that's been kind of historic, that when COVID came, even though people didn't think Moderna could do these things, really in 2 days after the Chinese scientists published the virus genetic sequence, they designed the vaccine, then put it in the nanoparticles, shipped it to NIH for testing, and the first patients were treated on March 16th, and this got approved even for use all over the world. And the next slide just shows a picture of the vial, and the next slide just shows some of the data, that it was 94% effective. Just for reference, flu is about 40% effective. And the only two vaccines in the United States that would get approved were messenger RNA and nanoparticles, using some of the principles that we developed. The next slide just shows really, again, why you want to get a vaccinated. It makes an enormous difference in terms of hospitalizations. And the next slide just shows that this is not an easy thing. This was really in less than a 2-year period, there were 49 vaccines that failed and only 2 approved. There were 209 treatments that different companies tried to make and only 1 approved. And with antivirals, 153 failed and there was only 1 approved. Nature would write that different people would write, if we could go to the next slide, that really this 1976 invention, and of course, as I mentioned, continued by many great people in our laboratory over the years, including David Putnam, would really raise the possibility of making this as a drug. And not only can it be used for COVID and other regular vaccines, but what's really exciting is it may open the door to cancer vaccines. And that's going to be shown on the last slide that I want to go over, which is really what Moderna and Merck are doing, is using this to treat patients with melanoma. Here you see 157 patients with melanoma, and what we're doing is making a personalized cancer vaccine. That's done by selecting up to 34 mutations known as neoepitopes in the patient's cancer cell. And you take their genetic code and you do exactly what you did for COVID. You just incorporate that genetic code into the mRNA in nanoparticles. In the trial, half of the patients, and it's double blind, get Merck's drug Keytruda, which is probably the best immunotherapy drug, and half get Keytruda plus the personalized cancer vaccine. And what you see is at 2 years, the risk of recurrence or death was reduced by 44%, and at 3 years, reduced by 49%. So I want to end here, but just make a few comments that, to me, invention has been a wonderful thing. It's very interesting when I look at what's happened with even the whole area of mRNA. It's just 2 little companies, Moderna and BioNTech, that took the risk because of the different work that people did and made messenger RNA therapeutics, which really, I think, changed the world but by these vaccines, and I think will continue to do so. No large company would get involved until Pfizer finally in 2020 started working with BioNTech. No one was willing to work with us, which was probably ended up for the better in the sense of what's happened with Moderna. But so much of this has had to do with invention, with patents, and I just hope you have a wonderful day today, at this invention celebration. And I know from my experience at Cornell the wonderful work that everybody does. Thank you so much.